Group 9.
Good morning, everybody. My name is Lucas Ortiz, and along with my group members Giovanni, Marin, Paul, and Mitch, we're going to be talking about the fate of the male race and trying to answer the question whether or not the Y chromosome is disappearing. There's been a lot of research about this, and there's a lot of controversy, so we'll try to use some scientific evidence to convince you of our opinion. To do this, we must start with our character Larry. Larry is your typical male with one X and one Y chromosome. It has long since been determined that the Y chromosome is in fact what makes Larry a male. Currently, however, the Y chromosome is 1/3 the size of the X chromosome, which has sparked much scientific debate regarding whether or not the Y chromosome will be lost altogether. We are here today to talk about the importance of the Y chromosome, whether Larry will lose the Y chromosome, and if he does, whether it's important in the first place. In order to understand the evolution of the Y chromosome, we must go back to the time of Larry's ancestors, approximately 300 million years ago, when the X and the Y evolved from autosomes and began diverging. It was at this point that the Y chromosome started its degradation. That stopped 25 million years ago. And now the Y chromosome contains 3% of X chromosomal genes. So the Y chromosome went from 1,500 to 50 genes due to inversions, deletions, oxidative stress, and translocations. These genes remain lost due to selective pressure and the inability to recombine with the X chromosome due to these inversions. Because of this inability to recombine, Y chromosomes kept on being deleted, leading to the ultimate degradation of the Y chromosome. So now that we know how the Y chromosome has been deleted, we must question why. The Y chromosome used to be composed of much junk DNA and repeat sequences. Evolutionarily, this junk DNA was not important at all to the organisms, so it was selected against. To put it simply, we did not need these sequences in the genome back then, and we still do not need these sequences in the Y chromosome today. 25 million years ago, the Y chromosome decay of the previous 275 million years halted, leaving Larry with an arguably stable set of ancestral genes. It's important to remember that at this time, tail-bearing monkeys were evolving into the ape and human lineages. Although Larry and his ancestors are far apart evolutionarily, there is little known variation between their sex chromosomes over time. So now we must ask, why does anything remain on the Y chromosome? Well, the Y chromosome currently has 50 active genes that remain due to dosage sensitivity, which means that any genes on the Y chromosome must have a counterpart on the X chromosome. Dosage sensitivity is so important because it makes sure that there are 2 copies of a given gene, which allows for their function. And the Y chromosome is not only important in sex determination, but also in gene regulation. For regulatory processes such as protein synthesis, gene activation, and RNA splicing. An example of this gene regulation comes with histone lysine demethylase in DNA remodeling, which neutralizes the positive charges on histone tails, causing histone slippage and opening up the DNA from heterochromatin to euchromatin, allowing transcription. So the Y chromosome, while it has many regulatory functions, it also codes for sex-determining genes such as the SRY gene in males, which codes for maleness by creating testes-determining factor, or TDF. TDF is a transcriptional regulator that essentially codes for maleness and makes us male. You can see the SRY gene on this chromosome, on this Y chromosome right here. So these Y chromosomes, specifically the SRY and EIF2S3Y genes, were proven to be able to create sperm cell precursors in mice called round spermatids through round spermatid injection, or ROSI. ROSI is a technique where we take round spermatids, implant them into female eggs, and create an embryo that can then be implanted into an organism to create a live offspring. So using ROSI with spermatids, we got a 9% success birth rate with mice, whereas while using just the Y chromosome in a similar technique to ROSI, there's a 26% success live birth rate. The fact that any organisms were produced at all from these procedures shows that we only need a few genes from the Y chromosome that we have now to make live offspring. So relating ROSI to humans, ROSI can help infertile men as a technique for infertility for men whose testes only produce spermatids. However, there's much debate surrounding this issue because it's known that, that this could cause genetic defects in the offspring possibly. Furthermore, we don't know exactly which Y chromosome genes are necessary to create a live offspring. So what makes a male and a female a female? Well, both male and females could have XX and XY cells in their body, which makes sex determination difficult. Furthermore, androgen insensitivity syndrome is a syndrome where you have the genotypic XY male, but you express as a phenotypic female, which adds further ambiguity to sex discrimination, showing that it's not quite as simple as it is here. So relating back to Larry and the fate of his Y chromosome, we do believe that his Y chromosome is here to stay. The Y chromosome stopped diverging, we found evidence of humans approximately 200,000 years ago compared to the 26 million years that the Y chromosome has been stable, showing that we're only a little piece of this evolutionary history and that the Y chromosome has been around for a long time before Homo sapiens evolved. So now that we know the Y chromosome is stable, we must ask whether science will be able to artificially create males. So even if the Y chromosome does disappear, we would have to look to the lonely X chromosome to find genes that were recombined onto the X chromosome from the Y chromosome a long time ago. Although we think that the Y chromosome will not disappear, we do believe that modern science will be able to restore the male race. Thank you.